Hello, my name is Laurel Brigham, and I'm a fourth year PhD candidate at CU Boulder in the Department of Ecology and Evolutionary Biology. Today, I'll be telling you about some of my research in the Colorado Front Range taking place at Niwot Ridge. And this research was generously funded by the Indian Peaks Wilderness Alliance.
This project focuses on how the expansion of shrubs might influence the range of subalpine plants. Shrubs around the globe are moving into alpine and Arctic areas at rapid rates. Importantly, these shrubs look quite different from the prostrate herbaceous plants you find in the alpine. Shrubs are taller and wider and thus create their own microclimates across the landscape. Shrubs trap snow during the winter and spring, they provide shading during the summer, and they offer wind protection. Here's a picture of a shrub from the area I work trapping snow in late spring. Additionally, shrubs impact soil nutrient availability and soil organic matter content. These soil effects result from leaf chemistry and from the large quantities of leaves dropped relative to herbaceous alpine plants. Together, these factors may create more benign conditions that support the growth of other plants, such as subalpine species. With global warming, some subalpine species will find themselves in peril if they are unable to track the changing climate by moving uphill. Shrubs in the alpine may act as stepping stones for subalpine species to start this uphill movement. To better understand the role that shrubs may play in subalpine range movement, I established plots at Niwot Ridge in the summer of 2019, made possible with funding from the Indian Peaks Wilderness Alliance. With help from 2 undergraduate researchers, we planted thousands of seeds in open plots and plots protected by shrubs. We also removed neighboring species from half of the plots to determine whether neighbors would impact establishment. See my Indian Peaks Wilderness Alliance video from last year for an in-depth look at the experimental setup. While we planted several subalpine species, germination was only high enough for analysis for Erigeron glacialis and Deschampsia cespitosa. We monitored germination along with soil temperature and soil moisture in 2019, but there was additional data that we needed to collect this year in order to fully understand the patterns we were detecting. Therefore, in the summer of 2020, I also collected information on the amount of incoming solar radiation, and I collected soil to measure soil pH and soil organic matter. I once more monitored the seedlings, and then at peak biomass in July, I harvested all seedlings. In the lab, I separated the above ground and below ground biomass. The above ground biomass was sent to the drying oven to be subsequently weighed, while the roots were stored in ethanol and would later be stained to determine root fungal colonization. First, I will outline the abiotic effects that shrub presence had at our site. Shrubs did not alter soil pH, but they did increase soil organic matter, showed higher soil moisture at peak biomass, and decreased the amount of incoming solar radiation. This confirms the presence of shrub microclimates at our site. Second, I will describe the treatment effects on Erigeron and Deschampsia germination in 2019. We will look at 2 different aspects, whether or not germination occurred, and given that it did occur, we will look at the number of germinants. Soil moisture had a positive effect on Erigeron germination. We saw that there was a soil moisture threshold, around 35% required. Additionally, germination was slightly more likely to occur in plots protected by shrubs. This could have resulted from microbe-mediated interactions, as we know that soil microbial communities differ between shrub-protected areas and open tundra. In terms of the number of Erigeron germinants, shrub presence had a positive effect. Higher soil moisture and protection from the high levels of solar radiation in the alpine are likely the environmental factors behind this finding, and microbes could play an additional role, which will be explored with the stained roots that I have harvested. We also monitored germination of a common alpine grass, Deschampsia cespitosa, which we seeded in the same plots. We used this comparison to determine whether there is a species-specific effect of the benefits of the shrub microclimate on germination and establishment. We found that the removal of neighboring plants negatively influenced whether Deschampsia germinated, but that shrub presence did not play a role. In terms of the number of germinants, we found that shrub presence only had a positive impact when neighboring species were removed. It appears that the likelihood of germination was increased when the plots were less exposed, but in the presence of shrubs, the number of germinants increased when there was less competition from neighbors. While the alpine species we studied required the removal of neighboring alpine plants, often the same species as itself, to experience the benefits of shrub presence on the number of germinants, our subalpine species performed better regardless of the presence of neighbors. Together, these findings suggest that the conditions created by shrubs may be beneficial for the establishment of subalpine seedlings with similar preferences to Erigeron glacialis.